Hello, everyone. My name is Ayesha, and over the past few months, I've been working with my amazing mentor, Sunday, and the Lumiere team to basically research how CRISPR can be used to treat skin cancer at a genetic level. So basically, what this means is cutting the part of the DNA that is causing cancer. And through this research, I amalgamated two of my interests, which are genetics and biomedical science. So the three main core questions that kind of guided my whole study. The first one is, how does skin cancer develop at the genetic level? So where does skin cancer start in the DNA? How does skin cancer start in the DNA? The second one, how does CRISPR work as a gene editing tool? Basically, the mechanisms of CRISPR and how it works. Third one, can CRISPR be used as a targeted and effective treatment for skin cancer by editing cancer-causing mutations? So I discussed the pros and cons, and additionally, either can CRISPR be used in clinical settings in real life, not just in experimental labs. So before we get into the actual study, I just want to give you guys a little bit of background of this topic and why is this important. So skin cancer, it is one of the most common cancers in the whole world. And it is caused by something as basic as sunlight. So UV radiation, it mutates the genes such as TP53, BRAF, NRAS, and many more. And they transform healthy normal cells, into tumor cells. Um, and the problem is that conventional therapies or conventional treatments, such as chemotherapy or surgeries, they remove the tumor cells, but they don't target the genetic cause. They don't target the mutations that is causing cancer, and thus cancer can come back at any point. Here's where CRISPR comes in. It is a powerful, precise gene editor because it targets the mutations. It's some mutations that are causing the cancer. It targets the root cause of the disease. And how it does that is shown in this diagram on the right-hand side of the slide. So basically, the DNA double helix, it contains a specific target gene. And the guide RNA, the orange strand, which you can see right here, it is designed to match the target sequence, and it directs the CRISPR-Cas9 enzyme, which is the green structure you can see, to the correct location. And once it is bound, it cuts both strands of the DNA at the target site, and this allows for gene editing, either disabling a gene, removing a mutation, or even integrating a new genetic material. So methods and analysis, what I actually did for my research. It wasn't an experimental research, so I started by conducting a literature review using peer-reviewed articles from Google Scholar and PubMed to basically establish a foundation and a background, which I mentioned in earlier slides. Secondly, I, along with my mentor, obviously, identified gaps and ethical concerns across existing research. Because the concerns, the gaps, the failures of CRISPR, it's just as important as the potential of CRISPR. And then finally, I kind of integrated my own thoughts and perspectives into the paper to make a detailed research paper which addresses the core questions which I mentioned in earlier slides. So now let's get on to the actual findings of my research paper. So CRISPR works, at least in the labs it does. And it's been used to target a lot of skin cancer genes such as TP53, BRAF, NRAS, and many more. So I already explained the mechanism of CRISPR in the earlier slides. So CRISPR targets specific mutations in cancer-causing genes, either by restoring normal function, silencing harmful mutations, or disrupting pathways that promote uncontrolled cell growth and survival. So CRISPR, it has shown a lot of success. It is very precise, and it has been called the best gene editor tool to this day. Why? A, because it is less expensive than the previous gene editing tools. It's expensive, but it's less expensive than the previous gene editing tools. And B, because it is more precise than the previous gene editing tools. But CRISPR isn't perfect. It faces major challenges. A lot of research still needs to be done in order to make CRISPR working tool in clinical settings. So some of the major challenges it faces includes off-target effects, which means it can sometimes miss its target. It can trigger immune responses, and delivering a CRISPR-Cas9 safely to the targeted site, it's still very complicated and it's still very risky. Now, the ethical question. Will CRISPR only remain available to the high income individuals? Because we're not denying CRISPR is going to be expensive. But in order to make it accessible for all, governments and scientists, they need to work together in order to make new policies to make CRISPR affordable and available to all. So that's it. To conclude, CRISPR is a new shift in the medical field, in the biomedical field. It just doesn't treat cancer. It basically rewrites the code that is causing cancer. So I hope you found these slides interesting, and I hope you learned something new. If not, I just hope you acknowledge that science can be powerful. Thank you so much for listening. Bye.